GLP-1 experimentally is it's broken down very quickly, and fortunately it was discovered that there's a compound, Extendin-4, it's derived from the saliva of the Gila monster, and it's a full GLP-1 receptor agonist compound. It's got a lot of structural homology, and it's not degraded by DPP-4. So it remains in circulation for about a 6-hour period. So this is a nice tool to be able to work with to get a better idea about what the potential for some of these compounds could be at reducing food intake. Two companies came up with compounds that were related to this that have now been approved for diabetes treatment, and one of them has just been approved as an anti-obesity treatment. We've demonstrated in non-human primate experiments that Extendin-4 is very effective at reducing overall food intake. And this is, this was data from rat experiments, so it's quite effective in that design. But in non-human primates that are consuming many more grams per day, you get a very nice dose suppression in food intake with increasing doses of this kind of Extendin-4. This is using a paradigm where the primates have access to food for a 6-hour period per day. So you can, with large enough doses, you can essentially completely prevent food intake in this kind of short-term situation. Taking a mid-range dose here and giving it over successive days. Here's the baseline intake. Giving the compound just prior to the daily feeding period, you get a consistent suppression in overall food intake across days, giving this kind of compound. When you stop giving the compound, they return right to the baseline. So these calories are essentially lost. The animals are not compensating for what these caloric deficits, and that's very different from if you didn't give the compound, but you pair-fed a group to what these animals had been consuming, and then stop that. The pair-fed animals will become hyperphagic, trying to make up for what that caloric deficit is, where the ones who got the Extendin-4 are not compensating for those lost calories. We were interested in understanding what the mechanism for that lack of compensation is, and so for these experiments we went back to the rat. What we were doing here was looking at the ability of this compound to affect food intake across days in conditions where the animals were either consuming standard chow or a high-fat diet. And what you can see is that in both situations here, the Extendin-4 is effective at reducing food intake across days. One of the interesting aspects here is, again, we had a pair-fed group here to get a view for just what that caloric deficit meant relative to what the suppression is. We were surprised to find that in the chow situation, the animals getting the Extendin-4 and the pair-fed group were different from a body weight standpoint. So the animals getting the Extendin-4 and chow were compensating for what those lost calories were in terms of changing what their energy expenditure is. So they were not losing as much body weight as the pair-fed. When we were giving them the high-fat diet, though, it was exactly the opposite. So there was something about the high-fat diet that was preventing what this kind of a compensation could be, and we don't really have a good understanding for that. We wanted to look to see how the Extendin-4 over time was affecting hypothalamic systems that are involved in the long-term control of energy balance. And there's been a circuit that's been identified in the arcuate nucleus of the hypothalamus that contains neurons that express peptides that stimulate food intake. These are called orexigenic peptides. They're NPY and AGRP, and a neuronal system that produces compounds that suppress food intake. So our questions are, what was our exendin treatment doing to gene expression in these two neuronal populations that might explain why there's not compensation? So I'm going to be showing you a series of graphs here that's going to be in this kind of a format, looking at the expression of various of these peptide systems. And on the left side of it, it's going to be the animals getting chow. On the right side of it, the animals consuming the high-fat diet. The animals consuming the high-fat diet consume more calories, first of all. So in response to consuming more calories, it's been shown previously that that dysregulates what this POMC signaling is, so there's no compensation in the baseline condition here. When we give the exendin 4 over that kind of prolonged period, in both cases, there's an overall effect of exendin 4 to increase what the POMC expression is. So it's activating this hypothalamic system in a way that it's going to suppress food intake. And that's not what's happening in the pair-fed situation. So this response is not due to what the caloric deficit is, but seems to be driving it. In contrast with the orexigenic signals, there's once again an overall effect at reducing signaling in this pathway, so reducing the expression of a brain peptide that would stimulate food intake. And again, you don't get that in the pair-fed situation. And we find a similar pattern with the other peptide that stimulates food intake. In the exendin 4 situation, the gene expression of that peptide is significantly reduced, and it's not occurring in the pair-fed situation. So changes in the signaling of these peptides, reducing the expression of peptides that would be stimulating food intake, increasing the production of peptides that would inhibit food intake, is most likely what's accounting for these changes in food intake and the lack of compensation. Because there's a real difference in what the signaling is. These two groups of animals are consuming exactly the same calories, but it's only in the exendin 4 situation that the gene expression is being changed. We looked at, as well in these experiments, at reward signaling, because others have demonstrated there can be a role for this compound in reward, and we really didn't find anything here. Similar to what's been demonstrated previously, consuming a high-fat diet really dampens what the reward signaling is, and the changes here seem to be all due to the high-fat diet rather than to the other manipulations. And the same kind of thing with one of the other aspects of reward signaling. In contrast to what we showed with CCK, using these brief access testing, once again, the exendin 4 reduced the licking for both sucrose and for the intralipid solution here. We think this is secondary to the fact that taste cells on the tongue express GLP-1 receptors. So one of the aspects through which GLP-1 is reducing food intake is by modulating taste. So it's got both a peripheral effect on taste and a central effect on hypothalamic signaling. So just to summarize, exogenous GLP-1 or GLP-1 agonists reduce food intake. They're unlikely to be involved in meal termination. Exendin 4 increases POMC expression and decreases the expression of arcuate peptides that decrease food intake. I'm not showing you some experiments looking at how the signaling from GLP to POMC is being mediated. There's no direct effect on reward signaling, but it does affect the consumption of palatable substances, and that's probably secondary to modulating taste. So we now know that we can reduce food intake with both of these peptides, and the question then becomes, is there a translational implication of this, and might we be able to get greater effects on overall food intake and body weight if we combine and activate these two systems simultaneously? I've shown you previously that using Extended 4, we can get very nice reductions in food intake. CCK over a daily 6-hour period really gets compensated for. So there's a brief suppression of intake, but that's not lasting. However, you can modify what the peptide structure is of CCK, maintaining its ability to interact with CCK receptors, but reducing the ability of proteases to break it down. And that's what this compound is. And so giving that compound over time, you can have a significant effect on daily 6-hour food intake. That led us to go forward with two compounds. One, a specific CCK1 receptor agonist compound that's got a variety of substitutions that result in a hexapeptide. It's got great specificity for the CCK1 receptor, so we're targeting that receptor population rather than just giving CCK itself that could activate both CCK1 and 2, and then a modified GLP-1 agonist compound that again is more resistant to degradation. These experiments are being done in rhesus monkeys, and with both of these compounds, you can see dose-related decreases in overall food intake using a paradigm where the animals have access to food for 6 hours a day. When you combine them, so what I'm showing you here is one dose of the GLP-1 analog and increasing doses of the CCK, you get a very nice suppression of food intake. So this dose by itself really has very little effect, but when you combine it with the CCK, you get suppressions, or you can view it from the opposite standpoint. Well, no, here's a larger dose that by itself produces a suppression, but when you add doses of CCK, you're increasing what that suppression is. Interestingly, when you look at these in combination, the combination dose is maintaining aspects of both of the compounds given individually. So the CCK compound is having its main effect at the beginning of the feeding period, and that's maintained with the combination compound. The GLP-1 compound has some effect, but its main effect is later during the feeding period. So these two lines are essentially parallel. You're not getting the kind of increase you get with the CCK compound by itself. So you're getting an increase in what the overall suppression is and maintaining the characteristics of the two individual peptides. We've gone to another non-human primate model. So these are genetically obese bonnet macaques. They were developed in a colony at Downstate run by Len Rosenblum, and through selective breeding, he could produce animals that essentially weigh 3 times what the normal animal does. And so if we are going to be affecting food intake and having an effect on body weight, would these compounds be also effective in these very obese animals? So here's just one experiment giving the combination across a 9-day period. There's variability here, and there's variability in the degree to which individual animals continue to have their food intake suppressed here. So these are kind of preliminary experiments. We're now undertaking an experiment where we're going to be giving this kind of combination for a 2-month period to see if we can sustain these kinds of suppressions and produce significant decreases in overall body weight. The effect of that combination is in the order of about a 40% reduction, and the whole phenomena is mediated by decreasing the size of individual spontaneous meals. So what we're producing here is earlier satiety using this kind of a dose combination, because there's no change in what the daily meal number is. Interestingly, using this 6-hour paradigm, these animals consume about 3 meals per day, so it's a nice model for what the human condition could be. We've been interested in how this combination, where in the brain this combination might be expressed in terms of suppressing food intake. So here's just a rat experiment, again using this kind of a dose combination. What we did in one of these experiments was to sacrifice the animals 2 hours into the feeding period and look at CFOS expression once again. I showed you how CCK itself, you can see it with these black dots here, induces CFOS expression at this hindbrain site. The GLP-1 by itself really doesn't do very much, but when you combine GLP-1, it really synergizes the degree of hindbrain activation produced by a dose of CCK here. So it may be that the combination here is producing the kind of suppressions in food intake through this kind of increase in hindbrain activation. Even though we've demonstrated that the GLP is working from a hypothalamic site, there are direct descending projections from the hypothalamus to this hindbrain site, and that may be explaining what the sensitization is here and why we're getting this great increase. So just to, I think I've got one more slide. Yeah, I do. Just to overall summarize here, both CCK and GLP-1 receptor agonists produce dose-related suppressions of food intake. The pattern of intake suppression differs. Combinations result in additive intake reductions, maintaining the characteristics of both compounds. We're now in the midst of experiments looking at whether longer-term administration of these can have a translational benefit in terms of overall reducing food intake and body weight, and whether that reduction can change what some of the consequences of obesity could be. None of these experiments, of course, get done by just one person. A lot of the work that I've been talking about today has been done by past colleagues, by postdoctoral fellows and graduate students, and again, colleagues here. And the work, all of the work, has really been supported by this one long-term grant here from the NIDDK. Thank you for your attention.
Thank you, Dr. Moran. We'll have some few minutes for questions, if any.
Can I?
Yes.
Dr. Moran.
Thank you very much, sir. It was very enjoyable and very informative. I just wanted to know that we are all sensitized by the CCK.
Yes.
So noble and graduate on this call. CCK. I have a couple of questions.
Sure.
The first question is pertaining to that the transmission, that how the CCK is transmitted through the afferent nerve fibers.
Yes.
And what is the mode? Are there receptors in the afferent fiber at the distal end?
Yes, so we've demonstrated that CCK receptors are expressed on vagal afferent cell bodies in the nodose ganglion, which is the major cell body for the afferent vagus, and they're transported along the vagus to these peripheral terminals.
So the peripheral terminals
Have the CCK receptor present, yes.
But then it has to be, eventually the information has to go through the hypothalamus. And that's what controls the feeding center. The information from those sensory ganglions goes through the hypothalamus.
So there's a bit of controversy on that. And so decerebration experiments where you're separating the hindbrain sites onto the nucleus of the solitary tract, where these vagal afferents terminate centrally from the hypothalamus. In that condition, CCK is still effective at reducing food intake. So it does not require hypothalamic activation. So for a lot of these compounds, the hypothalamus is really a coordination site that is taking into account what the fat stores are, what the circulating nutrients are, and that's providing a descending signal onto the hindbrain, modulating the degree to which these ascending signals are going to be effective.
My second question does not pertain to your